Uh, Thank you. Good afternoon. My name is Dagmar Szkornika, and it's my great pleasure to present you our company called Stimuliver. So a new startup that develops a disruptive implant technology to treat chronic diseases. So we are the biotech spin-out from the University of Edinburgh, and currently we are based at the Bioinnovation Institute in Copenhagen. And for the last 3 years, we've been supported by different Danish and international investors, and so far we have raised around 3 million euros for our preclinical testing and CMC planning. So our research is mainly focused on pluripotent stem cells, but we are interested in the liver as it's a fascinating organ. But unfortunately, liver disease kills approximately 2 million people per year worldwide. One of the most affected groups are patients who suffer from acute and chronic liver failure. And this syndrome affects about 200,000 patients per year and comes with a very high mortality within a month. And currently, liver transplantation is the only efficacious treatment for this particular group, but as we know, this approach is severely limited by organ donation. So we believe in taking a radically different approach to support these ACLF patients with a simple solution that doesn't require the simple solution that allows to supplement this liver function. Our aim that I'm going to describe in a moment is to generate a remote and off-the-shelf 3D liver tissue implant to top up patients' reduced liver function. So according to the Back Bay report from 2022, the total addressable market for ACLF is about 200,000. And however, it is predicted it will rise to about 300,000 by 2036. And Stimuliver, and we know this is very important across 4 major European markets, UK and the US. And Stimuliver mainly focuses on ACLF patients in grade 1 and grade 2 with a CLIF score below 70 that represent about 80% of the total ACLF population. And these patients usually have up to 2 failing organs, and they have a higher survival rate than group 3. So in order to overcome all these issues, we have developed a unique stem cell-derived liver tissue implant that may serve as a short-term treatment option for patients with ACLF. So more specifically, we differentiate pluripotent stem cells to 2 liver cell types, that is to hepatic progenitors and endothelial cells. And these differentiated cells are then combined and scaled up through our automated procedures to create a Then this liver tissue is loaded on an FDA-approved polycaprolactone scaffold and implanted beneath the patient's skin to begin filtering the toxin. So how it works? Once deployed underneath the skin, the 3D liver tissue implant displays a critical liver enzyme function and secretes a variety of proteins that recondition as circulating blood. And in this way, our implantable approach permits waste product detoxification, reduction of liver damage markers, and hence improves the structure of the host organ. So far, during our research at the University of Edinburgh, we have successfully differentiated pluripotent stem cells to 3D hepatocytes using a microwell differentiation in at least 4 stem cell lines available at clinical grade. And by using this system, we are able to obtain hepatocytes with appropriate morphology, gene expression, as well as with improved polarity, which is here indicated by the Z01 marker. What's more, our hepatocytes actually maintained a metabolic activity for at least 1 year in culture in vitro. So this potentially allows us to create a stable and ready-made liver tissue implant that can be maintained in the lab until required. So following this in vitro data, we have generated proof of concept in mice. So our 3D hepatocytes composed of polycaprolactone scaffold were subcutaneously transplanted into immunocompromised mouse models of tyrosinemia type 1, so to a model which is commonly known as FAH. And in this model, mice normally develop a severe liver toxicity. However, the accumulation of this toxin can be prevented by anti-BC drug. So in our studies, the 3D graft connected with the host blood circulation within 3 days and improved our recipient's liver function for at least 14 days in vivo. The implant displayed a critical liver support, leading to significantly reduced liver damage markers and improved clearance of toxins without any sign of graft rejection or degradation. What's more, to prove liver recovery in this mouse model, we demonstrated an improved host liver histology with our implant when compared to the empty scaffold that served here as a control. So the murine livers from the control group actually displayed swollen hepatocytes, apoptotic cells, and also multi-segmented nuclei. And these processes were, of course, less obvious in our experimental group. So following from these studies and observations that our graft is not rejected in animals with the intact immune system, we were interested in better understanding graft-cell interactions. So in collaboration with King's College London, we demonstrated that our hepatocytes possessed immunomodulatory properties. So when hepatocytes were incubated with T cells, they did not activate T cells, but in fact inactivated them. And this was due in part to the lack of MHC class II and co-stimulatory molecules expression on hepatocytes. In addition, hepatocytes also express IDO1 enzyme, which metabolizes tryptophan. And this tryptophan starvation of CD4 and CD8 T cells actually resulted in reduced proliferation and activation, even if the T cells were pre-activated before co-culture. So all these positive results led us to, let's say, further modify our established differentiation protocols, and we decided to form liver spheres, this time by combining hepatic progenitors with endothelial cells at the ratios found within the liver. And of course, such refinement would allow us to further improve the hepatocyte proliferation and also hepatic vasculature. So since we were able to successfully differentiate stem cells to hepatic progenitors and endothelial cells in 2D, we then wanted to test whether we could generate liver spheres. So by using our, let's say, chemically defined protocols, we were able to successfully aggregate these cells and maintain them for at least 6 weeks in vitro. And these liver spheres displayed a healthy, opaque, non-necrotic phenotype with the potential endothelial tubules extending from the center of the spheres, which is here indicated by the red eye. And of course, this suggests that our new spheres may vascularize more quickly and also more efficiently connect to the blood circulation in the future. We also observed that the immature markers such as AFP decreased within 6 weeks, and this decrease was achieved 8 weeks faster than in our previously published data. And moreover, albumin and alpha-1 antitrypsin, so the mature markers, remain stable over time. As we know, scale-up and scale-out are immensely important in our field, and therefore we have developed uniquely scalable manufacturing processes to generate prototype liver tissue with improved liver functions. And up to now, we have, let's say, established standard operating procedures to provide a consistent spheroid formation from research and clinical lines. So by using our, let's say, defined automated pipetting system like ViaFlow and custom-made 96-well plates, we are now able to generate about 7,000 spheres per plate. And all these spheres, they display a similar size. They also express key liver markers such as HNF4 alpha, ikatherin, and vicatherin, as well as they demonstrate an increase in metabolic function for over 4 weeks. I would like to mention that this system has been tested for 2D and 3D models, and it's highly reproducible for at least 10 pluripotent stem cell lines, including ES and iPSCs. So once these cells are scaled up, we place these cells on a polycaprolactone scaffold to stabilize the phenotype, metabolic function, and also to prevent any cell migration. And here, this is just the example of our initial graft, which, let's say, displayed the potential vascularization after 2 months in vivo, and no additional issues were actually observed. Currently, we assume that the patient may require about 50,000 spheres per one tissue implant in order to top up their failing liver function in the future. So at the moment, we are in a cycle of manufacturing, optimizing, and implanting our liver tissue. Post-specification and prior to transplant, our liver tissue is usually characterized by multiparametric analysis. And this, of course, includes biochemical, proteomics, metabolomics analysis to quality assure our liver tissue. And following this, the liver tissue is implanted in vivo, and its, let's say, performance and durability is studied in small animals with large animal studies planned for the near future. And these in vivo studies or results will help us to further refine our tissue product for clinical studies. So in summary, our concept called remote regeneration comes with several advantages. We have developed potentially safe and remote 3D liver tissue implant from a renewable and sustainable source of pluripotent stem cells. Our scalable and off-the-shelf 3D liver spheres offer a solution to top up patients' reduced liver function that may delay liver transplantation or promote regeneration. What's more, our solution doesn't require complex surgery and also avoids a hostile environment of the liver. The implant could be delivered to patients using a local anesthetic, and no tissue rejection would be observed. So combined, we provide a treatment which is available without waiting time and has the potential to prevent hospitalizations. So thank you very much for your attention. I would like to thank our team and also our scientific advisory board for their fantastic support, as well as the organizers for the opportunity today. So thank you again, and I'm happy to take any questions. Of course, albumin, but also from my experience and my PhD, these are also the drug metabolizing enzymes. So in case of cytochrome P450 enzymes, that would be 2D6 enzyme, also CYP3A4, and of course, phase 2 enzymes, so sulfotransferases and glucuronosyl transferases, mainly UGT1A1. Thank you.